All right, let's dive into something pretty incredible today, the field of bioinformatics. We're talking about the science that's teaching us how to read and maybe, just maybe, one day even write the book of life itself.
Let's just kick things off with a really bold statement. Back in 2003, J.D. Tisdale said, if you can't do bioinformatics, you can't do biology. I mean, that's a powerful claim, right? And it perfectly sets the stage for just how essential this field has become. It's not just another tool in the toolbox, it's become the actual language of modern biology.
So, what exactly is this thing that's so indispensable? To really get what a bioinformatician does, we first have to understand the problem they were basically born to solve.
You see, the whole field came into being because of one massive, overwhelming problem, a complete and utter tsunami of data. In the last century, biology started generating a staggering amount of information.
Think about it. Huge undertakings like the Human Genome Project opened the floodgates, unleashing this torrent of information. It got to the point where biological databases were doubling in size every 15 months. It became totally impossible for any single scientist, or even a huge team of them, to manually sift through all of that data.
To really put this into perspective, just look at this number, over 4.6 million. You know what that is? That's the number of base pairs in the genome of a single E. coli bacterium. Now just try to imagine scaling that up to the human genome, which has 3 billion base pairs. The data problem wasn't just big, it was astronomical.
And a massive problem like that, well, it needed a totally new kind of solution, and you could say a new kind of hero for the data age. It was going to require merging the world of biology with the raw power of computer science.
And that solution was bioinformatics. It's the science of using computational horsepower to make sense of all this biological information. The key thing to remember is that it's an interdisciplinary field. It's a bridge between life science and computer science, and it was built specifically to manage, analyze, and interpret that data tsunami.
And you absolutely cannot tell this story without talking about Margaret O. Dayhoff. She's often called the mother of bioinformatics, and for good reason. She was a true pioneer. We're talking way back in the 1960s, she started painstakingly collecting all the known protein sequences into something she called the Atlas of Protein Sequence and Structure. She was applying computational methods to biology before that was even really a thing.
Dayhoff's atlas back in '65 really kicked off an explosion. Fast forward to '82, and the public gets access to GeneBank, which is this massive sequence database. Then, a huge leap in 1990, the BLAST algorithm came out and completely revolutionized how scientists could search for similar sequences. Just 5 years after that, we sequenced the first complete genome of an organism. The momentum was just incredible. Okay, so now we know why bioinformatics exists. But what does a digital biologist's toolkit actually look like? How do they turn all that raw data into genuine discoveries? Well, the process usually follows a pretty clear path. It all starts with compiling and storing the data in a really disciplined way. Then comes the fun part, the analysis, where you're looking for meaningful patterns. But hey, the tools are never perfect, so a huge part of the job is actually developing new algorithms. And ultimately, the real goal is to integrate all these pieces to understand the entire biological system, not just its individual parts. Honestly, the best way to think about these tools is with a simple analogy. The massive databases are like global libraries, and the algorithms, well, they're the super powerful search engines for those libraries. Together, they let scientists find one tiny piece of information hidden among trillions of data points. And these aren't just, one-size-fits-all libraries. No way. They are highly specialized. So if you're looking for the 3D structure of a protein, you'd head over to the PDB. Researching a specific genetic disease, you check OMIM. Need a raw gene sequence? GenBank is your go-to. Each database holds a different type of clue for solving these huge biological puzzles. Okay, so what's the real-world payoff for all this digital detective work? This is where all that abstract data becomes something incredibly tangible. Life-saving drugs. This right here is the core principle of so much of modern medicine. If you can use bioinformation to pinpoint the exact gene or protein that's causing a disease, and then you can understand its structure and how it works in detail, well, then you can design a drug that specifically targets it. It's like having the enemy's blueprints before you even design your weapon.
And the applications are just astounding. This is what's ushering in the era of personalized medicine. We can now diagnose genetic disorders with incredible accuracy, predict a person's vulnerability to certain diseases, and even develop targeted cancer therapies that attack tumors while leaving healthy cells alone. This isn't science fiction anymore. This is modern medicine, and it's all powered by bioinformatics. So we've seen the problem, we've met the pioneers, and we've seen the incredible impact. So what's the big picture here? What does the road ahead look like? I think the main takeaway is this. Bioinformatics has grown up. It's evolved from some niche specialty into an absolutely essential discipline. It is the engine that turns raw biological data into actual knowledge, allowing us to truly understand the code of life and use that understanding to improve our world. And that leaves us with one final fascinating thought. For decades, we've been totally focused on reading the book of life. But as we get better and better at reading it, we're getting closer and closer to being able to, well, write in it. And as we master that ability, the great question of our time becomes not just what can we do, but what should we do? What will we choose to write next? Thanks for tuning in.